studies that correlate drug treatment responses in patients with drug treatment responses in iPSC-based models of those particular patients. I think there's some work in this area trying to correlate sort of clinical trials that are happening in patients with clinical trials that are happening parallel in a dish with iPSC-based models of those individuals. So, I'm really excited about the future and what we'll see in this area over the next few years.
Welcome to the Personalized Medicine Podcast. This is the place where scientists, clinicians, and entrepreneurs discuss the progress of this rapidly developing field. I am your host, Oleksandr. Let's start. Three, two, one, and we are live. Welcome to the next episode of the Personalized Medicine Podcast. In this episode, we are returning again to the topic of stem cells. This time, we will look at their role in drug discovery. It is my pleasure to introduce to you Professor Evangelos Kiskinis. Evangelos is an assistant professor at the Northwestern University Feinberg School of Medicine in Chicago. He completed his PhD at Imperial College London and then moved to Harvard to do a postdoc at the lab of Kevin Eggen, where he was harnessing the utility of stem cells to study and treat neurodegenerative disorders. After starting his own research group at Northwestern in 2015, Evangelos has been focusing on developing iPSC-based models for various neurological conditions, including ALS and pediatric epilepsy, and his work also led to exciting clinical trial in this space. Evangelos, thank you so much for accepting my invitation and welcome to the show.
Great. Thank you for having me. I'm excited to be here, and I appreciate the invitation.
Perfect. Evangelos, I would like to start with your story. Can you tell us what got you interested in stem cells and how that passion led you to the place you are at today?
Right. So, just a little bit of background. I'm originally from Greece. I grew up in Greece, the youngest of three, and I first got intrigued and excited about science around high school, where a talented biology teacher sort of illustrated to me that the power of science and biology, and how when you study something you discover a new world. So that's when I first became passionate about science. I moved to the UK when I was 18, and I did my undergrad and grad school there. And during grad school, I worked on cellular reprogramming and how metabolism can alter the function of cells. And it was around that time that Shinya Yamanaga published a seminal paper in Cell, where he described how you can take 4 transcription factors and turn any adult somatic cell into a pluripotent stem cell, which shares the capacity to differentiate into any cell type in the human body. So I read that and I got really excited. I think it became increasingly apparent to me how transformative this potential technology could be. So I started reading more and more about it. And at that point, I decided that that's what I want to do for my postdoctoral research. So I emailed a few people. I emailed Kevin Eggen, who was a pioneer and leading iPSC-based research at the time. And I said, look, we've never met before, but I'm really excited about your work. I'm really passionate about it, and I want to come and, work for you. And he said, great, come on over. And so that happened in 2008. And that was sort of my first foray into the stem cell world. I was in Kevin's lab, where I focused on understanding, the properties of induced pluripotent stem cells and how we can utilize them to study and understand neurological diseases. When I left Harvard, and started my own lab here at Northwestern, I knew that I wanted to stay in sort of around the same theme of using stem cell based technologies to model and try and understand how neurological disease develops and whether we can use these approaches to halt or restore degenerative processes in human cells.
Perfect. Speaking about neurodegenerative disorders, those conditions are extremely hard to treat and also to understand on the basic fundamental level. What do you think makes induced pluripotent stem cells such an exciting model to study neurological diseases?
Right, that's a great question. So, I think, as you've said, as you mentioned, it's been really hard to, model neurological diseases. I think for a couple of reasons. I think, firstly, there's a lot of diversity within the central nervous system, and there's a lot of human-specific properties in terms of both different subclasses of neural and neuronal subtypes that exist, as well as the function of different cell types within the CNS. And that renders modeling these diseases with traditional animal models such as mice, rats, fish, flies, and so on and so forth challenging. So I think that's been one issue, and the other one is that the central nervous system is fairly inaccessible. It's really hard to access the cells that are degenerating within the brain or the spinal cord of people that suffer from these diseases. And if you can't access the cell type that's primarily affected, it's really hard to study and appreciate the disease. So I think iPSC technologies have really offered a workaround around this, or a solution to this accessibility issue. And they also provide us with an additional model, which is based on human cells, which carry the unique genetic makeup of each particular individual. Mutant or disease proteins are expressed under physiological control systems in the right cell type and in the human setting. So I think they're a useful tool that can be used in parallel to all the other animal model systems that have been traditionally used in the study of neurological diseases.
Perfect, understand. And developing those models is not an easy task. Also speaking a little bit from my experience as a PhD student, what are the main challenges in establishing a good model based on human iPSC cells for those neurological conditions?
Right. Very good point. I think there are several challenges that the field has been facing and continues to face. I think, having said that, we've made tremendous progress in bettering the model systems that we create with iPSC technologies, and I think we've certainly have come a long way from sort of the first generation iPSC-based models of neurological diseases. I think we've become better at differentiation protocols. We've become better at making different neuronal subclasses, making the right subtypes. We've become better at making more sophisticated systems that incorporate multicellular aspects of diseases with not just neurons, but additional neuronal subtypes, glial cells, and so on and so forth. I think the organoid work that has been happening has really facilitated the generation of this next level model systems. But there's certainly several challenges that remain. I would say, if I had to break it down, I think there are three. I could probably distill it to three areas. The first one is relates to the fact that iPSC-derived neuronal cells seem to resemble neuronal subtypes late embryonic or early postnatal stages of human development. And when you're trying to understand an adult onset or late onset neurodegenerative disease like Alzheimer's or even ALS, that can be a challenge. So I think figuring out a way to mature and in fact age the cells that we make, the neurons that we make from stem cells, is an important challenge. I think we've already seen a few breakthroughs around the area related to direct reprogramming, for example, of adult somatic cells directly to neurons, which seem to retain some of the aging factors of the parental somatic cell and the patient, as one exciting development. But I think we'll see, I know that there are several other developments in the pipeline related to manipulating the factors in vitro to age iPSC-derived neuronal cells. I think we're going to see some of these come out in the next, in the imminent future. So I would say aging is one challenge. I think the other one relates to what I talked about a little bit, which is the multicellular aspect of several diseases. So we know that it might be, for example, dopaminergic neurons in PD or motor neurons in ALS that are primarily driving the disease, but we also know that there are several other subtypes that contribute to disease, and I think we have to move towards developing multicellular model systems. And again, the organoid technology that has been that's being developed and becoming better and better, it's going to solve a lot of these problems. And I would say that the third area is the fact that iPSC models are in vitro models of disease, and when you're thinking about diseases like epilepsy, for example, it is a disease of the brain. It is a disease of an intact nervous system, and so that is a challenge. We always have to keep that in mind. But again, I think the organoid work that has been that has been developing in the last few years is going to address some of these challenges. I can particularly think of some of the phenomenal work that has come out of Sergio Pasca's lab at Stanford, where he's successfully recreating an entire network, with upper motor neurons and lower motor neurons and muscle as one example, or GABAergic interneurons synapsing and with excitatory neurons. These are really exciting developments, and I think we're going to see breakthroughs in this area over the next few years as well.
Fantastic. We are doing this show for you, and your feedback is very important for us. So if you have any suggestions or comments, would like us to cover a specific topic or recommend a guest, please write us an email to team@pmedcast.com. Or you can reach out to us on LinkedIn, Twitter, or Facebook. Just type in Personalized Medicine Podcast and you will find us there. To download the show notes for this episode, visit our website, pmedcast.com. It's pmedcast.com. The show notes include guest bios, links to their most notable work, and recommendations for additional reads on the topic of the episode. Make sure to check them out. And don't miss the next episode. Subscribe to our podcast on your favorite podcasting platform. Give us a rating and leave a comment. It will help us make this show better. And now, let's get back to the interview. neurological disorders, it's a pretty broad group of diseases, and some of them have some underlying genetic causes behind them. Some of them don't, or at least seem not to have. How does that impact our ability to model those disorders in the dish with iPSC cells?
Right, that's a very good question, and it's a real. I think it's related to your previous question. What are some of the challenges that we're facing? So obviously, I think genetic neurological disease or neurodegenerative disease is probably the first sort of disease that we have been tackling as a field, and the reason for that I think is because I think that it is rational to think about what are the best ways of controlling for some of the phenotyping you're doing when it comes to genetic disease. And I think here's where the editing technologies that have been developed over the last few years with CRISPR specifically, most recently, allows us to test the idea that it is specific mutations that cause particular phenotypes that we identify in a dish. And I think CRISPR technology has allowed us to basically, correct a mutation in case we generate an iPSC line from a particular patient that we know has a particular disease-associated genetic mutation, and then ask whether correcting that mutation restores a particular phenotype from a iPSC differentiated neuronal subtype. Well, we can also do the opposite. We can introduce a disease-causing variant in a iPSC line derived from a healthy genetic background and ask whether that disease-causing variant is sufficient to introduce a phenotype. So these are critical controls that have enabled iPSC based modeling labs to really create ask this critical questions, and I would argue are becoming a standard in what's expected from an iPSC model these days. I think where we still have a challenge is sporadic disease, which unfortunately represents the overwhelming majority in most cases of neurodegenerative disease like Alzheimer's and ALS. And I think sporadic disease is likely caused by a combination of genetic predisposition, probably polygenic contribution, variants that contribute of low penetrance that combinatorially lead to increased susceptibility to disease, play a role in sporadic disease as probably the interaction with the environment. So that's whether or not epigenetics also contributes to this process is up for debate. It likely is, and that's where iPSC technologies face a challenge, because work over many years ago has shown that when you actually create an iPSC line, you wipe out almost entirely the epigenetic profile of the adult somatic cell. So if epigenetics does play a role in sporadic disease, then that will render iPSC models problematic. A solution here could be these direct reprogramming approaches that have been described by several groups over the last few years, which seem to maintain some of the epigenetic profiling. So I think that's one issue that we need to tackle as a field. But I think if it's not just epigenetics, and I think another challenge that we have is how do we control for sporadic iPSC based disease modeling? What are the right controls we should use if we want to model sporadic disease in a dish? And I think there's some exciting developments that are hopefully we'll see in being published soon with sort of multiplexing technologies, which solve a problem of bandwidth and sort of studying multiple sporadic diseases and multiple sporadic iPSC disease lines and multiple control iPSC lines at the same time. So I'm looking forward to seeing some of this work, and I think that's where that will be the next sort of generation of modeling that we'll see going after sporadic disease as a critical um, breakthrough that we need to see happen.
Perfect. Yeah, and absolutely. It's a challenge, and we don't have any shortage of challenges, specifically with those sporadic diseases. But let's hope that cumulatively we can advance science and then also our scientific insights to clinics to tackle those. And speaking about advancing science into clinics, can you tell us a bit about your work on the ezogabine trial for ALS? And what were your key takeaways from that project?
Right. Yeah. Thank you for asking this. So I guess the backstory here is that in work that I did a few years ago when I was a postdoctoral fellow in Kevin Egan's lab and in collaboration with Brian Wanger and Cliff Wolf at Children's Hospital at the time, we developed preclinical models of ALS, different genetic subtypes of ALS, and we uncovered as a group that one of the salient features of these that was present across different genetic subtypes of ALS neurons was this disturbances or disruptions in the excitability pattern of ALS motor neurons. And we also through sort of irrational small scale screen of a select number of drugs, we discovered that ezogabine, a drug that targets potassium channels, specifically Kv7 channels that are abundantly expressed in motor neurons, seem to effectively restore some of these excitability deficits that were present in the ALS motor neurons. And I think we published our work, and it gained some traction, and in work that was continued by my colleague Brian Wagers and others. That preclinical model discovery led actually to a phase 2 clinical trial for ezogabine for patients that have ALS. So I think the first point that I'd like to make, it's an exciting milestone, the fact that a basic discovery in a preclinical iPSC based model led to a clinical trial for that particular disease. And the clinical trial was completed. It was a phase 2 trial that included 125 patients. The result of that trial was published in JAMA Neurology a few months ago, and it basically showed that ezogabine was tolerated in ALS individuals. And it also showed that treatment of ALS patients with ezogabine restored the cortical hyperexcitability that was recorded in these individuals. So it met its primary endpoint. So that was the second milestone as far as I'm concerned. I think the trial was not designed in a way to assess efficacy properly. It was a short-term treatment for 12 weeks, and so the hope is that this work will be followed up by doing a larger sort of efficacy based trial. Right. So that that I'm excited about some of the work, and in my own lab we're continuing a little bit to try and understand a little bit more about what is the mechanism behind the excitability deficits that are leading to that are apparent in iPSC derived neurons, and why is Zolga been particularly effective at restoring these?
Got it. And now speaking about stem cells, not just as model for studying neurological diseases, but rather as a potential agent to actually treat those disorders. We've seen quite a bit of progress in cell replacement therapies in Parkinson's disease over the last few years, but not so much still in ALS or Alzheimer's. What are the main reasons for that, and what, as a scientific community, we have to do to make sure that we can also use differentiated neurons as a potential therapeutic option for those conditions?
Right. Yeah, that's another great question. So, as you rightly pointed out, I think there's excitement in the field of cell replacement therapy. That there's a few clinical stage companies that are engaging in that. Blue Rock Therapeutics is I think one of the companies that's focusing on cell replacement for Parkinson's disease by making stem cell derived dopaminergic neurons. And I think other exciting diseases in this area are probably related to neuroimmunology with microglia, and potentially we'll see some exciting developments in that area, or oligodendrocytes and demyelinating disorders. I think that the fundamental difference between these areas and something like ALS or Alzheimer's is, I think in Parkinson's disease, we know the cell type, and we know the specific, which is the dopaminergic neuron, and we know that this exists in a very specific area in the brain that's that can be accessible, I guess. So the task of replacing is not as formidable as in ALS, where not only do you need to deliver the motor neurons in the spinal cord, but you need to rely on those cells to sort of extend their very, very long axons outside the central nervous system and synapse with muscle, in the periphery. And that is a major challenge that we face as a field. And so that's, I think, one of the primary reasons why we haven't seen more movement in the area of cell replacement, particularly for ALS. Having said that, there is some preclinical work around ALS and cell replacement, or transplanting, neural cells, that not specifically motor neurons, and neural cells that could be beneficial. I think we'll see the results of those trials soon. But I think it's a little bit more complicated than Parkinson's disease. I think I'd like to point out that there's been a focused effort, sort of creating dopaminergic neurons that are de facto dopaminergic neurons that do most of the things that neurons are known to do from stem cells. There's been also fantastic preclinical work in sort of transplanting these in mouse models and other preclinical models. So I think that's been the focus for many years, and that's how we're seeing an impact for that disease, for cell replacement. I think for other diseases, because of the challenges that I mentioned, there hasn't been that concerned effort in achieving cell replacement for now.
Got it. Yeah, it takes a long time to plant the seed until you can harvest something, specifically in those neurological diseases. But staying a little bit more positive and kind of speaking about the future. So what I'm curious about is to get your outlook on what do you think the three major developments would be over the next decade on this interface of the stem cell biology and neurological disorders?
Right. That's a good question to think about. So, I think the first thing that comes to mind is I think as a field, we really need to solve the issue of aging iPSC-derived neuronal populations to sort of model and recapitulate robust neuropathology, to robustly recapitulate that in vitro model systems. And I think, again, that we'll see breakthroughs in this area. So I think that'll be crucial. I think, I would like to see, iPSC models becoming a standard part of assessing, the mechanism as well as, efficacy of drug development programs. And I think, again, I think this is, we're in the cusp of basically it's happening. I think there are several biotechs and pharma companies that are increasingly adopting stem cell based models in their preclinical programs. Testing, for example, specificity and efficacy of technologies such as antisense oligo technologies for genetic subtypes of diseases, or CRISPR-based therapeutics. So it'd be nice to see more of that happening. And I think the other thing that I can think about is that we really need to see more and more examples of preclinical work based on iPSC-based models leading to discoveries of therapeutics that are actually being applied or tested in clinical trials in patients. Like we saw with the zogabine, I think there's more of those examples in the pipeline. And if I can be allowed to mention a fourth thing that I'd like to see, which I also think is in the pipeline, is studies that correlate drug treatment responses in patients with drug treatment responses in iPSC-based models of those particular patients. I think there's some work in this area trying to correlate sort of clinical trials that are happening in patients with clinical trials that are happening parallel in a dish with iPSC-based models of those individuals. So, I'm really excited about the future and what we'll see in this area over the next few years. I'm really excited to see those studies being coming out.
Yeah, and I completely agree with you. And I really like this last point that you mentioned. It is extremely important for personalized medicine just to understand how individual patients respond to specific drugs, and then can iPSC-based models derived from those patients actually behave similarly. So that's a very interesting point.
Yeah, I agree with you on that, and I think this is, we're doing some work in this area with epilepsy models, and we're truly trying to understand whether our models are going to be good enough to sort of predict how a patient would respond to a particular drug. And obviously we're not the only ones. I think there's several groups that are engaged in this type of work, and several biotech companies. So be exciting to see the results of those efforts.
Absolutely. Perfect. Evangelos, you've had such an inspirational career so far, and you've been working on the cutting edge of neurological disorders, stem cells. Which one advice could you give to young scientists who are listening to this podcast and who are thinking about continuing or staying in the academia for long haul and perhaps work in the stem cell research?
Right, that's a good question. I think The first thing I like to say is that there's never been, I think, a more exciting time to engage in this type of research. I think some of the technology that's being developed, some of the breakthrough, organoid-based work that we're seeing coming out, some of the translational aspects of stem cell-based models, that are being developed. It really is an exciting time. So, I think in terms of advice, I think, the first thing that comes to mind is be bold. Be bold. Don't be afraid to reach out to people. And engage with them and talk to them about why you're excited about the work that they do, about sharing some of your ideas and your perspectives, some of the things that you want to achieve in this area. I think not a lot of things will happen if you sort of are reserved and sort of quiet and don't reach out to people and don't seek opportunities. So be bold and grasp opportunity that comes about around you. Engage with other researchers in this area and sort of turn your ideas into reality. Test them out. Don't leave any stone unturned. If you have a good idea, make it happen.
Perfect. And I'm sure there are a lot of bold people in our audience who perhaps would like to reach out to you. Where can they find you online?
Yeah, so I think where they can find me on our website, which is Kiskinis lab at northwestern.edu. They can also ping me on LinkedIn, where I'm active and I sort of post developments from my lab, or I comment on exciting developments from lots of other people. So I'm always looking to be engaged and talk to other people. So yeah.
Fantastic. Evangelos, thank you so much for this interview. It's been extremely exciting to speak about this booming field of stem cells, and specifically in this very hot application area of neurodegenerative and neurologic disorders. I'm sure our audience will love it, and we have a lot of neuroscientists who are tuning into this podcast on a regular basis. So thanks a lot, and we'll stay in touch, and all the best to your lab, and looking forward to seeing more wonderful research coming out of it.
Right. Thank you. Thank you for having me. This was fun. I appreciate it.
Thank you so much for being with us today on the Personalized Medicine podcast. If you like this show and know someone who would enjoy it too, please share this podcast with them. And don't miss the next episode yourself. Subscribe to the Personalized Medicine podcast on your favorite podcasting app. You can find us on Apple Podcasts, Spotify, Pocket Casts, Stitcher, iHeartRadio, and many, many more. Please rate us there and leave a comment. That helps us to grow and deliver the best experience to you. To access the show notes for this episode, visit our website, pmedcast.com. It's pmedcast.com. And engage with us on social media, where we regularly share the news and exciting content on personalized medicine. You can find us on Twitter, LinkedIn, and Facebook just by typing in Personalized Medicine Podcast, or use our handle, pmedcast. And if you have any feedback or would like to suggest a guest for the show, write us an email to team@pmedcast.com. Have a great day, and until next time.